Okay, so welcome back to this next video in which we are discussing voltage-gated ion channels. Okay, so we're currently in the process of discussing voltage-gated calcium channels, and we've discussed that the main subunit of a voltage-gated calcium channel, the principal subunit, the one that actually has the pore down the middle through which calcium ions are capable of moving, is called the alpha-1 subunit of the voltage-gated calcium channel. Okay, we've discussed how similar this is to the alpha subunit of voltage-gated sodium channels. In fact, the basic membrane spanning topology of these alpha-1 subunits of voltage-gated calcium channels is exactly like that for the voltage-gated sodium channels, and indeed the way it folds up, okay, and where the different domains contribute is exactly like we saw for the alpha subunits of voltage-gated sodium channels. Now, we've been discussing then all of the different types of alpha-1 subunits of voltage-gated calcium channels then. Okay, so we've seen that there are 10 different genes for alpha-1 subunits of voltage-gated calcium channels, and that they're grouped into these three different families, the CAV1, CAV2, and CAV3 families. And we've been discussing how there are these other names for the different types of voltage-gated calcium channels that are produced from alpha-1 subunits of these different types. So we've discussed that L-type voltage-gated calcium channels, when someone talks about an L-type voltage-gated calcium channel, all that means is that the alpha-1 subunit of the voltage-gated calcium channel in question is from this CAV1 family of alpha-1 subunits. So it was produced from one of these four genes here, CAV1.1 to CAV1.4. Okay, we've also discussed PQ-type voltage-gated calcium channels and how they are voltage-gated calcium channels where the alpha-1 subunit is CAV2.1 type alpha-1 subunit. N-type voltage-gated calcium channels where the alpha-1 subunit is CAV2.2 type. Okay, R-type voltage-gated calcium channels where the alpha-1 subunit is CAV2.3 type. And then finally, T-type voltage-gated calcium channels where the alpha-1 subunit is within this CAV3 family. Okay, right, so what I now want to talk about, now that we've discussed the basic structure of the main principal subunit of a voltage-gated calcium channel, is I want to go over the basics. I want to go over voltage activation, permeability and selectivity, okay, and then talk about inactivation. And then we'll go to something new, which is all of the different accessory subunits that voltage-gated calcium channels can have. And then we'll talk about some drug molecules that can block voltage-gated calcium channels of the different types. Okay, right, so we'll start off with voltage activation. So it's the same old story that we're getting bored of now, okay? These alpha-1 subunits are incredibly analogous, we have seen, to the alpha subunits of voltage-gated sodium channels, and indeed the gating mechanism is the same. So the reason that the channel is closed in the resting state when the electrical potential difference across the cell membrane is, let's say, negative 65 millivolts, is because the S6 helices, these ones in red here, are all coming into the middle of the pore on the intracellular aspect of the pore, so they're all converging to the centre at the intracellular aspect of the pore, and hence closing off the internal portion of the pore so that no calcium ions can move through there. Okay, so once again that is called the V-gate, and when you depolarize the electrical potential difference across the cell membrane up to threshold potential, then again these S4 membrane spanning alpha helices, which have got positively charged residues in, are going to move upwards, okay, because they're no longer feeling that same pull towards the intracellular compartment, and they will tug on the S4, S5 linkers, which then conveys the conformational change to the pore-forming domain, and the S6 helix will then move outwards, and you will then have opened the V-gate, and now calcium will be able to flow through the ion channel. Oh, excuse me. Okay, so that's voltage gating. Let me just add some further nomenclature into these different types of voltage-gated calcium channels. Okay, so the nomenclature that I want to now add in is the concept of a high voltage activated and a low voltage activated calcium channel. Okay, so calcium channels are divided into two very broad classes. Okay, HVA calcium channels stand for high, that's the H, and then the VA is for voltage activated. Meanwhile, LVA stands for low voltage activated, so the L is for low, and then these two words here are exactly the same. Okay, so these 10 different types of voltage-gated calcium channel alpha-1 subunits, they're divided up into these two very broad categories. Okay, so what's the difference between high voltage activated and low voltage activated voltage gated calcium channels? Well, the name gives it all away. High voltage activated calcium channels are the ones which it takes quite a hefty depolarization of the electrical potential difference across the cell membrane to actually activate. So they have quite a high threshold potential. You need quite a lot of depolarization from resting membrane electrical potential difference to get up to the threshold potential for a high voltage activated voltage gated calcium channel. Okay. Meanwhile, the low voltage activated voltage gated calcium channels, those require a much less hefty depolarization of the electrical potential difference across the cell membrane to get them to open. Okay, so which of these alpha-1 subunits are high voltage activated and which are low voltage activated? So voltage-gated calcium channels built from alpha-1 subunits in the CAV1 and CAV2 families, those are going to end up being high voltage activated voltage gated calcium channels. So if your alpha-1 subunit is from the CAV1 family or the CAV2 family, so it's an L-type or it's a PQ type, N-type or R-type voltage gated calcium channel, then your voltage gated calcium channel is high voltage activated. It requires a hefty depolarization of the electrical potential difference across the cell membrane in order to actually get it to open. Okay. Meanwhile, if your voltage-gated calcium channel is a T-type voltage-gated calcium channel, i.e. its alpha-1 subunit is from the CAV3 family, then that voltage-gated calcium channel will be low voltage activated. It doesn't require the same sort of hefty depolarization of the electrical potential difference across the cell membrane in order to get it to open. A much more feeble depolarization will do to get it to open. Okay, so you often hear these pieces of terminology, high voltage activated versus low voltage activated, so that's what that refers to. Okay, so that is the gating mechanism then of the voltage gated calcium channels, and it didn't take us that long because we've seen it all before basically. Okay, now let's talk about permeation and selectivity. Okay, again, this isn't going to take us that long because it is very similar to what we have discussed for voltage-gated sodium channels. Okay, so basically for voltage-gated sodium channels, we saw how the selectivity filter was lined by these 4 ascending p-loops from the 4 different domains of the alpha subunit of the voltage-gated sodium channel. In the case of the voltage-gated calcium channel alpha-1 subunits, it's going to be exactly the same. Okay, the selectivity filter is going to be lined by the 4 ascending p-loops of the alpha-1 subunit of the voltage-gated calcium channel. And again, this is quite a wide selectivity filter, much wider than we had in the voltage-gated potassium channels, and it's much less rigorous in how the ions are actually held in the selectivity filter, basically, than the voltage-gated potassium channel selectivity filters. Okay, and indeed, again, when calcium ions move through the selectivity filter of voltage-gated calcium channels, they do not give up their entire hydration shell. Okay, calcium ions also have a hydration shell of water molecules surrounding them, and when they move through the selectivity filter of the voltage-gated calcium channels, they're not going to give up all of their water molecules, basically. They're only going to give up some of them, okay, in order to interact with electronegative groups that line the selectivity filter, basically, and that's how it will move through in the same way that we've seen for how sodium moves through. So partially hydrated, basically, it will be in a partially hydrated state as it moves through the selectivity filter. Okay, and I discussed at the time when we were discussing the selectivity filter for voltage-gated sodium channels what it is that confers selectivity for selectivity for calcium in a voltage-gated calcium channel. In voltage-gated sodium channels, you just had a single ring of negatively charged residues, basically. Okay, so every single one of these ascending p-loops had at this same level either a glutamate residue or an aspartate residue, which are negatively charged amino acid residues, and therefore you had this ring of negative charge here, basically. Okay, and for alpha subunits of voltage-gated sodium channels, you only had one ring of negative charge. I keep trying to call it positive charge. You only have one ring of negative charged residues, okay, and that then confers selectivity somehow for sodium ions over calcium, whereas if you put an additional ring into the alpha subunit of a voltage-gated sodium channel, that turns it into having selectivity for calcium ions rather than sodium ions. And indeed, the alpha 1 subunits of voltage-gated calcium channels, they do naturally have two rings of negatively charged residues, basically, in their selectivity filter, giving them this selectivity for calcium ions, basically. Okay, right, so that's permeation and selectivity. That's how the calcium ions get through the selectivity filter. And then, of course, just like before, the internal portion of the pore is that wider portion of the pore where the calcium ion can move through it in the hydrated state. Okay, just like for voltage-gated sodium channels and voltage-gated potassium channels. Okay, so that's voltage activation and permeation and selectivity. Now let's talk about inactivation of voltage-gated calcium channels. Okay, so again, you have two different types of inactivation of voltage-gated calcium channels. You have fast type inactivation or N-type inactivation, and you also have slow type inactivation. Okay, so let's discuss fast type inactivation first. Okay, so just like with the alpha subunits of voltage-gated sodium channels, it is believed that fast type inactivation in the alpha 1 subunits of voltage-gated calcium channels occurs through one of those linkers. However, it's much more controversial in alpha 1 subunits of voltage-gated calcium channels which linker it is than it is in the alpha subunits of voltage-gated sodium channels. Okay, it is known for voltage-gated sodium channels that the linker between domain 3 and domain 4 is responsible for doing that hinged lid action and shutting off the intracellular entrance to the pore, basically. Okay, and thereby blocking any conduction through the channel. Okay, in alpha 1 subunits of voltage-gated calcium channels, it's much more controversial which bit actually mediates fast type inactivation. However, we think it is the 1-2 linker. This is the sort of best bet, basically. Okay, that it's the 1-2 linker. So the linker between domain 1 and domain 2, and we will be seeing more of the linker between domain 1 and domain 2 later on. So let me just bring back up my picture of what the voltage-gated calcium channel alpha 1 subunit looks like. So here is the voltage-gated calcium channel alpha 1 subunit. Okay, and here is the linker between domain 1 and domain 2. So here is domain 1, here's its voltage sensing domain, here's its pore forming domain. Here is domain number 2's voltage sensing domain, and here is its pore forming domain, and this here is the linker between the S6 helix of domain 1 and the S1 helix of domain 2. So it's that linker there, the 1,2 linker, that we believe can then bind to the inner opening of the pore and seal over it. Okay, and it will do that binding when the electrical potential difference across the cell membrane is depolarized of resting membrane potential, basically. Okay, however, again, it takes longer for this inactivation gate here to close than it takes for the V gate to open. So when you initially depolarize the electrical potential difference across the cell membrane, firstly the V gate will open and before the inactivation gate has closed, and therefore transiently you will actually get a pore that is conducting calcium ions. Then this inactivation gate, which we believe is probably linker 1 to 2, will then shut over the internal opening of the pore and therefore close conduction, basically stop conduction. Okay, but it's that time difference, that lag between the two, that then means that for a little flash, you are actually in that open state where you're conducting calcium ions through. Okay, and then if you want to now return back to the resting state where you're closed, then you need to repolarize the electrical potential difference across the cell membrane. Then what will happen is linker 1/2, the inactivation gate, will cleave off the internal opening of the pore. Okay, so you undo the inactivation, that's called de-inactivation, but of course also you will undo the opening of the V gate. The V gate will also now close because you have repolarized the electrical potential difference across the cell membrane. Okay, so you still will be in a closed state where there's no conduction occurring. Okay, so that is the fast type inactivation. We also believe voltage-gated calcium channel alpha 1 subunits have a slow type of inactivation, which again involves this voltage-dependent collapse of the selectivity filter, which blocks calcium from being able to move through it. Okay, but that again isn't very well understood. Okay, and again that will be reversed by repolarization of the electrical potential difference across the cell membrane. Okay, right, so that's inactivation then of voltage-gated calcium channels discussed. Okay, so now what I want to turn my attention onto is the accessory subunits of voltage-gated calcium channels, and this is a big thing now for voltage-gated calcium channels. There are 3 different types of accessory subunits that you can have joined onto the alpha 1 subunit of a voltage-gated calcium channel. Okay, and we're going to go through these 3 different types of accessory subunits and what they what their function is. Okay, right, so we'll start off by discussing the beta subunits of voltage-gated calcium channels. Then we'll go on to the alpha 2 delta subunits of voltage-gated calcium channels, and then finally we'll go on to the gamma subunits. Okay, right, so the beta subunits of voltage-gated calcium channels then. The first thing to say about the beta subunits is that there are 4 different types of beta subunit of voltage-gated calcium channel, and these 4 different beta subunits of voltage-gated calcium channel are called the CAV for voltage-gated calcium channel, and then beta for beta subunit, and then we've got CAV beta 1, CAV beta 2, CAV beta 3, and then CAV beta 4. Okay, right. Now, all of these proteins are non-integral membrane proteins. They're not integral membrane proteins. Okay, they are globular proteins that are on the cytoplasmic side of the membrane, and they're going to attach onto the bottom of the alpha 1 subunit of the voltage-gated calcium channel. Now, there is a specific portion that they attach onto, basically, okay, which is a portion that we've already met. Okay, they specifically attach onto a domain in the 1,2 linker. Okay, so let me now draw out the alpha 1 subunit of a voltage-gated calcium channel, and I want to put in a new domain this time, which I haven't shown previously because we were using the picture from the alpha subunit of the voltage-gated sodium channel. Okay, but what I'm now going to put in is a new domain that is specific for alpha 1 subunits of voltage-gated calcium channels and which wasn't present in voltage-gated sodium channels. Okay, but it's a minor difference, which is why it didn't matter that previously I used the picture from the alpha subunit of the voltage-gated sodium channels. Okay, right, so here is domain 1 here. Here's the amino terminus of the alpha 1 subunit for a start. Here is domain 1, and now there's this special domain in the linker between domain 1 and domain 2, which I'll give you the name of in a moment. Here then is domain 2, whoops, and I've missed off the p-loop, haven't I? How am I going to correct that up? Okay, so there we go, we'll have to colour it in to fix that. Okay, there's domain 2, here is domain 3, there's the p-loop, domain 4 here, okay, p-loop, and then there is the C-terminus over here. Okay, so I'll just colour this in to hide my terrible error there. Okay, so here in orange, this is domain number 1 here. Okay, here in red, this is domain number 2 of our alpha 1 subunit of our voltage-gated calcium channel. Okay, next up we'll have domain 3 coloured in green here. So this is domain number 3. Okay, and then finally we'll have domain number 4 here, which we'll colour in blue here. Okay, so there's domain number 4. Okay, right, so let's now turn to this interesting new box that we've got in the 1,2 linker here. Okay, so this special box is where the beta subunits of voltage-gated calcium channels are going to attach onto the alpha 1 subunits of voltage-gated calcium channels. Okay, and this special box is called the AID, the AID. Okay, and what does this stand for? Well, this stands for alpha, that's the A, interaction, that's the I, and then domain, that's the D. So this is the alpha interaction domain. Okay, so this is the interaction domain of the alpha 1 subunit that's going to interact with the beta subunit of the voltage-gated calcium channel, basically. So, voltage-gated calcium channel beta subunits are going to attach onto this alpha interaction domain here. Okay, and what's their function going to be? Well, they are going to alter the speed of inactivation, basically, which you might have predicted, okay, because we know that this 1,2 linker is probably the portion that is responsible for fast inactivation, okay. It's the portion that is going to perform this hinged lid role, okay, where it's going to close over the internal opening of the pore, basically. If we're going to attach something onto that 1,2 linker, then obviously that's probably going to affect the ability of the 1,2 linker to do its job of inactivating the voltage-gated calcium channel alpha 1 subunit. So these CAV betas are going to affect the speed at which the voltage-gated calcium channel inactivates, basically. Okay, right, so I want to now draw a three-dimensional picture where I show the alpha 1 subunit in those, in the sort of more true structure, how it really looks, rather than as this picture. And I want to then show you the CAV beta subunit attached onto the alpha interaction domain, basically. Okay, so I'm now going to draw this three-dimensional picture, which I think I'll try and squeeze in here. Okay, and I just want to make sure this is straight, yeah. Okay, so here, this is the pore of the alpha 1 subunit of the voltage-gated calcium channel. Okay, here is, well, this is the actual pore. This is the pore domain, basically, the pore-forming domain. Okay, here are the 4 separate domains that all contribute, the 4 separate pore-forming domains of the 4 separate domains of the alpha 1 subunit that all contribute to the pore domain. Okay, and then around the outside, of course, what we'll have is these voltage sensing domains of each one of the domains of the alpha 1 subunit of the voltage gated calcium channel. So here is another voltage sensing domain, another one over there, and another one here. So I'll just colour this in firstly, and then I'll try and add on to this picture the alpha interaction domain, and then I'll put on the beta subunit of the voltage-gated calcium channel. Okay, right, so let's firstly colour in a few of these domains. Oh, and I should have tried to colour code it with this, but I've forgotten the colour coding there. So here is domain number 1, which I'm now going to colour in red, so forget the colour coding there, basically. Okay, so here is domain number 1. We've been, throughout this video, calling this one closest to us the domain number 1, so I don't really want to now say, okay, we'll rotate round 90 degrees and call that one domain number 1. No, I'll just keep this one as domain 1 and just break with the colour coding. So this is domain number 1, here in red. Here is its voltage sensing domain. Here is its pore forming domain. Okay, and then we'll have domain number 2, here in blue, which is a nice stark contrast to red there. Okay, now we've truly broken with that colour coding over there. Okay, there's its voltage sensing domain of this blue domain here. Okay, and here is the pore forming domain over here. Okay, so the red domain is domain number 1, okay, of our alpha 1 subunit of the voltage gated calcium channel. So I'll just make this obvious. And then the blue domain is domain number 2 of our voltage-gated calcium channel alpha 1 subunit. So that means that we're going to have a loop down here, which is going to be the 1,2 linker here between the S6 helix of domain 1 and the S1 helix of domain 2 here. And this is the 1,2 linker shown here. Okay, and in that 1,2 linker, we are going to have this special domain called the alpha interaction domain. And this alpha interaction domain is then going to have a beta subunit potentially bound to it. Usually they do. Okay, so here now is our voltage-gated calcium channel beta subunit in orange here. Okay, and this beta subunit will be modifying the properties of the alpha 1 subunit. Specifically, one of the main ones that it modifies is how quickly the alpha 1 subunit will inactivate after opening, basically. And we can understand how that beta subunit might achieve that, since it's bound to the 1,2 linker, which is, we know, is probably very important in the fast inactivation of the alpha 1 subunit. Okay, right, so that's the beta subunit then of voltage-gated calcium channels, and you can attach one beta subunit onto one alpha 1 subunit then. Okay, next up I want to tell you about the alpha 2 delta subunit, so I think we'll try squashing this in here. So alpha 2 delta subunits next. Okay, so again, there are 4 different genes known for alpha 2 delta subunits, and I'll explain exactly why they're called alpha 2 delta subunits in a moment. Okay, so there are 4 known genes for alpha 2 delta subunits, and these are called CAV alpha 2 delta, okay, and then there is CAV alpha 2 delta 1, CAV alpha 2 delta 2, CAV alpha 2 delta 3, and then CAV alpha 2 delta 4. Okay, and basically let me start off with the alpha 2 delta portion. Okay, so basically these genes, these 4 genes, the CAV alpha 2 delta genes, they will code for proteins which have 2 portions in. Okay, so let me show you the protein that they'll produce. Okay, so each one of them will produce a protein like so. Here's the amino terminus, here's the carboxylic acid terminus, and then at the start of this protein, you will have a portion that is going to make the alpha 2 portion. Okay, and then you will have a portion that is going to make the delta portion. Okay, so each one of these genes makes a pro-protein, basically, which contains an alpha 2 portion and a delta portion. And then what happens is this is post-translationally cut up into the alpha 2 portion, which is at the amino side, and the delta portion, which is at the C-terminal side. Okay, and these two then are linked back together in a different way, basically. Okay, they are attached to one another again by a different way, so they're not linked back together by a peptide link. Instead, they're attached together by disulfide bonds. Okay, and that subunit that they then make which is a subunit of the voltage-gated voltage-gated calcium channels. An accessory subunit of the voltage-gated calcium channels is called the alpha-2 delta subunit, basically. Okay, right. So they're kind of two separate subunits, okay, because it's not just one polypeptide, it is two separate subunits, but because of the fact that they're made by the same gene, they are considered just one subunit, basically, okay, and they're called the alpha-2 delta subunit of the voltage-gated calcium channel. So the alpha-2 delta subunit consists of an alpha-2 portion and a delta portion, okay, which are not linked together by a peptide link but by disulfide links, but because they were made by the same gene, we consider them a single subunit. Okay, right, so let me now discuss the structure of alpha-2 delta subunits. Well, this used to be very simple. The way we used to think alpha-2 delta subunits worked is you had the delta portion, which was a transmembrane portion, and then attached to that on the extracellular side, you had the alpha-2 portion. Okay, so let's colour these in. We'll have alpha-2 coloured in here in green. Okay, and then we'll have delta, which actually I think I'll do in turquoise rather than blue. Okay, because I want to add it onto this picture, and I don't want it being confused with domain 2 here, which is in blue. Okay, so delta will now represent in turquoise rather than blue. Okay, so everyone used to think that this is how alpha-2 delta subunits were structured. The delta subunit was a transmembrane protein, and then on the extracellular side you had the alpha-2 subunit stuck onto it by disulfide links. However, the viewpoint is starting to shift towards a different picture, where delta isn't actually a transmembrane protein. Okay, instead it looks more like this. You have the delta here with the alpha-2 sat on top of it. Okay, so how does the thing then get attached to the cell membrane? Well, instead, the delta subunit gets a lipid anchor attached to it. Okay, so what happens is you attach onto the delta subunit a molecule called GPI, which I'll show here. Okay, this little yellow blob down there, that's supposed to be a GPI molecule. So where can I fit this in? I'll put it up here. So GPI. Okay, now GPI, its full name is glycosyl, that's the G, and then it should be all one word. Glycosyl phosphatidyl, that's the P. Okay, and then the I is for inositol. So glycosyl phosphatidyl inositol is what GPI stands for, and I'll just color code up this name. So the G is for glycosyl, the phosphatidyl is for P, okay, and the inositol is then in pink here. Okay, right, so the delta subunit of the alpha-2 delta subunit, or the delta portion of the alpha-2 delta subunit, has this, is believed to have, instead of having a transmembrane domain, is instead believed to have a GPI molecule attached to it. And basically a GPI molecule is just a very fancy phospholipid. It's just a really fancy phospholipid that can be part of a phospholipid bilayer. So basically you attach onto the delta subunit a phospholipid that then anchors into the phospholipid bilayer as a normal phospholipid. Well, not as a normal phospholipid, but as a phospholipid, okay? And then holds the entire alpha-2 delta subunit here on the extracellular aspect of the cell membrane. Okay, so I don't know if there has been a fundamental conclusion on this yet as to whether this is the correct one or this is the correct one, but for a very long time this was the one that everyone believed, whereas now it is starting to shift, as far as I'm aware, to maybe it's just attached to the cell membrane by a GPI molecule. Okay, whatever the truth is, the alpha-2 portion is certainly extracellular, and it's attached to the delta portion, which is either transmembrane or it's attached to the cell membrane by a lipid anchor, like so. So let me now show you how alpha-2 delta subunits attach onto the alpha-1 subunit of a voltage-gated calcium channel. So here we've got our picture of the alpha-1 subunit with a beta subunit there. Okay, what I now want to do is add on to that picture the alpha-2 delta subunit, and each voltage-gated calcium channel alpha-1 subunit can only have a single alpha-2 delta subunit actually attach on. Okay, and it's going to attach on in this gap here between the voltage sensing domain of domain number 2 here and the voltage sensing domain of domain number 3 here, and next to the pore forming domain of domain 1 and the pore forming domain of domain 2 here. Okay, so let me now put this in. So here is the delta subunit here, and then the alpha-2 portion is going to be up here. And I shouldn't say the delta subunit, I should say the delta portion of the alpha-2 delta subunit. Okay, here is the alpha-2 portion then of the alpha-2 delta subunit, sort of stretching over the top of the alpha-1 subunit there, and there is the delta portion there in turquoise of the alpha-2 delta subunit. Okay, now the alpha-2 delta subunit is believed to do a huge number of different things for the voltage-gated calcium channel. One of the things that it's supposed to be really important in is deciding the location that the voltage-gated calcium channel ends up in. So it's believed that it combines to a whole bunch of other proteins and hence localize the voltage-gated calcium channel in a certain portion of the cell membrane. Okay, so location is one of the big things that alpha-2 delta subunits are involved in controlling. Okay, it's also known that they can affect the threshold potential for the activation of the voltage-gated calcium channel alpha-1 subunit. Okay, and it's also known that they can actually affect also the speed of inactivation. So these alpha-2 delta subunits can affect a lot of things: location of the voltage-gated calcium channel alpha-1 subunit, the threshold potential for the alpha-1 subunit of the voltage-gated calcium channel, and also how long it takes to inactivate once it's actually opened. Okay, right, so we'll call it there for this video, and then in the next video what we'll do is talk about the final type of accessory subunit of voltage-gated calcium channels, which is the gamma subunit, and then we'll just talk about some drug molecules and some toxins that are notable for blocking certain types of voltage-gated calcium channel.